Hi, sorry, I'm gonna go a little out of order for this video. This is the part one of our essay for unit 3. I'm going out of order, so I'm sorry, because I thought I recorded a video, but I didn't, so I have this still up here. I don't want to erase it. So, when in our cell we have action potentials, those action potentials are going to be switching the charge of our ICF and our ECF in our cells. Normally, ICF's negative, ECF's positive, but we switch that when we are doing a heart contraction because we are trying to, unit 2, we have our muscles, they're pulling on each other, stretching, they're relaxing, all that good stuff. We have positive when we have our heart. Here's like our top right, I would guess. We have our negative electrode up here and a positive electrode down here. When we have a vector, it's a vector. We have a vector that is propagated by a negative turning, or a positive turning into a negative, or something along those lines, and it's going towards a positive electrode. We would see something like this. We'll have a positive slope. But if we see the opposite and it's going towards negative, so like going towards like, we would see the inverse of that. That out of the way. Let's get into PQ, our S and T waves. For P waves, we have our heart. These are our ventricles, these are our atriums. We have our SA node and our right atrium. I believe that's right, yeah. It starts our depolarization of our cells, and we have our cells that were positive now become negative, and this spreads through the cells. It's really confined to these two atriums. It doesn't go down into the ventricles. It does not pass. So, what did we just talk about? We have negative vector going towards a positive electrode. That is going to produce a little hump, like this one right here. Now for the QRS waves complex, I guess you could call it, it's a little bit more complicated. You're gonna have to use your imagination because I'm not very good at drawing. So for our QRS wave, we have two things that start it. We have our bundle of His and our AV node. AV node is more secondary than anything. It's mainly just bundle of His. And I think our Purkinje fibers play a role in that as well. But regardless, we have a depolarization. They start at the top, and that sets a negative vector to a negative electrode. That produces an inverse wave. That's what we get here. This is the Q wave. And once that's hyperpolarized, we start moving our way down, and we fill in all this over here, and we fill in this side last. So the filling in of everything except for right here is what gives us this big peak right here. It happens very fast, and it spreads a lot. That's why it's so big. But here, when we're filling in our last little bit, this is where you have to use your imagination. I'm not very good at drawing. So normally the vector would be more kind of up towards the negative. Now it kind of just looks like it's in between, but it's not, I promise. This is a negative vector, and it's pointing closer to our negative electrode than it is to our positive in the real life. Not here, because I drew it bad. But that would give us our S wave, and that's why it's an inverse wave, or it's inverse. And then we have completely polarized our ventricles. And similar to with our P wave, we don't go past the dividers between the atriums and the ventricles. Lastly is T waves. T waves are pretty simple. It's kind of just it's just a little bit of a different way. So all of this, I'll draw on this point, I promise. So starting at that last step, this whole thing is polarized. It's all negative, but we wanted to get rid of that because we don't like it just staying there. It's not like our P wave where it just goes away. So we are going to depolarize it. We are going to have vectors vectors that are going out this way, and these vectors are positive. So that's why we're going to get a little bit more positive one. And this is a little bit of a slower process. You work around from the edges, kind of. So we have a bunch of vectors that are pointing kind of everywhere. So that's why this doesn't get as big as our wave. It's smaller, it takes longer. It's not just a little boop, it's ooh. Once it's completely depolarized, like we have here, we end our wave. And that is the waves of the heart. I'm going to move into what I should have done first, which is our action potentials and cardiac ventricular muscles, SA node, and describing the channels, ions, channels, and membrane potentials that are found in all of those. Alrighty. So the first thing we need to know, there are two types of cells in the heart that we're going to talk about. We're going to be talking about autorhythmic and cardiomyocyte cells. Autorhythmic are for your kind of just your basic heart cell. Nothing too special about them. Well, there are special, but there's a lot of them. In our SA node, we have our cardiomyocyte cell. And we're going to start with our autorhythmic cell because it's a little bit easier to talk about. Autorhythmic cell, and it's very much the same thing as just a smooth muscle cell. It just has some extra nifty things. The big one of those is what we like to call our funny channel, but it's also just called the HCN channel. HCN channel is just a voltage gated channel, I believe. Once you have a big enough voltage, you're going to have an action potential enter, have a Bourn move down, and we're going to open up I'm going to put it here, a voltage gated calcium channel. Calcium is going to flood in, and we are going to it's going to trigger our sarcoplasmic reticulum, like in a regular muscle cell. And I believe it's our RYR receptor that opens these up in the cell. And that's just going to be a big channel, and a bunch of calcium is going to flow out, and it's going to form a contraction. Now, what makes these interesting is it's not like a muscle. You can't just go, I don't know, like, our heart beats a lot faster than that, and we don't have to think about it. So what makes our heart different in relaxation, we have uptake of calcium, just like any other cell. This goes back in. And to get rid of all that calcium so we can get another contraction going like that, we have these special channels. I just believe I'm not entirely sure, I do not quite remember what they're called, but they are special. They kick out of calcium because we can get calcium. We're going to get extracellular calcium no matter what, so we don't really care. It's fine if we care about stuff. Most of it's going to be reuptaken. We just want to get everything else out. So we have something that pumps out calcium and pumps in 3 sodiums. And this like drastically improves our sodium potassium pump. Ooh, sorry, I gotta sneeze. And that dramatically improves our sodium potassium ATPase. And what this looks like for an action potential is it looks something like this. I'm sure it's a bubble. These both end at the same place, and that place is -60 millivolts. We can kind of see everything that we talked about. We're starting here. We have a little bit of a net positive charge. Comes in, opens up our calcium, a lot of calcium, and then we start pumping it all out. And then we have to pump a little bit more out here. And then we repolarize to our resting membrane potential. Nothing too crazy. Now where the fun begins is with our cardiomyocytes. I think I said this before, but cardiomyocytes are found in your SA node. And they have a lot of receptors and a lot of channels. So I hope I get them all right. But we have a muscle cell, well, somewhat of a muscle cell. Just the way that it was written in the book. I don't think we need anything too crazy here. You can draw them with them or without them. Whereas our autorhythmic cell had the funny channel, we don't. We have a voltage gated calcium channel. When we reach it, get an action potential, that opens, we have sodium that floods in, and something similar to what happens in our autorhythmic cell happens. Action potential, boop, 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 goes down, opens this up. We have an influx of calcium, because this is a calcium gated voltage channel. Calcium goes here, and to our plasma reticulum, and we kick a bunch of calcium out. Now what makes this cell special is that we have some special receptors. They're not receptors, channels. They're ion channels, and this is where it gets a little messy, so I'm going to have my paper here that I wrote my notes on so I don't get these wrong. We have my two calcium channel. We have good voltage gated channel. Voltage gated. Okay. Sorry. This also has the calcium channel that was talked about. We have these 5 special channels. It's going to get really messy if I just try to say them all the time, so they're here. The action potential for this looks very strange. We start at -90 as opposed to -60. We have, that's kind of a big spike. Alright, this is what it looks like. So there's a reason why this looks different. We have these channels here because we need to get moving quick with cardiomyocytes. We need to be able to depolarize it and completely depolarize it. We have all of these channels here that help us do that, get us back to resting membrane potential. I'm going to draw some arrows. And hopefully I do this correctly. R2, R3, potassium these are all here to help us ensure that we get back to our true resting membrane potential. If we didn't have them, we could not do this. If there's a problem with one of these, that cell will not work. There's a reason that we have these, and they are there to help our SE fire. I should have done this backwards, but I had everything on the board for the first time I did this. So I decided to do it that way. I'm sorry if that didn't work. A big thing that plays a role in this, these cells are gap junctions and GERT channels. I believe that's mainly in autorhythmic cells. That's how you can get stuff moving. Gap junctions allow for everything except for water, I believe, to go through them. That's how you can kind of have cell cascades that happen. Yeah, you have Purkinje fibers in your bottom, bone day, or something branches, your cells. I believe that's the big things. So thank you so much. I'm sorry for the order that this has happened. Hi, good evening. I'm going to be doing the second part of my third essay, I believe, for BIOS 461. And I'm going to use a lot of colors. Let me just grab one real quick. Alrighty, got all my colors. And I'm going to be talking about pressure, time, and volume in the aorta, and stuff that's happening in the left ventricle as well, and throughout the heart. So these can best be described in two different graphs. The first graph is a pressure. Time graph. Use three different colors here. Our purple is going to represent our left ventricle. Our blue is going to be our left atrium. Green is our aorta. Okay, so we're also gonna I'm gonna draw this out and then I'm gonna explain what happens in it as we go. Yes. So for our left ventricle, you have a little bump in pressure, and then you're going to have a very big bump. Come back down. Go back to normal, and that'll be the beginning of another one of these. For atrium, have a little bit more pressure. But we're not going to have a pressure spike like with our ventricle. For our atrium, we're going to stay kind of up here. So there is a couple of big events that you can kind of see happen here. Here we have three different heart sounds, but we really only talk about two of them. First one's lub, the second one's dub. Lub happens here, and it is when your bicuspid valve opens. Oh no, sorry, it closes. It only makes a sound when it closes. Here, we have dub. And that is when your semilunar valve closes. Semilunar opens. And it's here we have a little thing, this little bump that's called your dicrotic notch. Yes, here this notch. where your atrium contracts. I'm gonna draw one more graph and we can go over it all. Next one's gonna be a little bit bigger. Pressure. Volume. Just over here. 60, no, 50, 20. Doesn't look straight, but it's supposed to be. Yeah, now this just looks like I'm drawing something, but it will make sense once I start. Here. Alrighty. So now that is all drawn out, let's start from the top. So this is going to be covering some of the mainly just pressure and time and certain compartments in your heart, your left ventricle, your left aorta, and your aortic arch. It's your aortic arch, this is your left ventricle, and this is your left aorta. Your left aorta doesn't really change much. It's not going to have a lot of pressure, whereas with your ventricle, you are going to have a lot of pressure because you are pushing that blood from your aorta kind of through your body. You're going to need a lot of pressure to do that. Whereas with your atrium, you're just pushing it into the ventricle, so you really don't need that much. Your aorta is going to stay about the same. You have to have high pressure throughout your body in order to have blood going places. If you don't, you're dead. So don't have low blood pressure, you're lying. Your diacrotic arch is pretty important. It's kind of just symbolizes where like blood pressure kind of really dips in your body, in your aorta, in your arteries. There's two sounds that we want to talk about, lub and dub. Lub is the first one that happens, and dub is the second one that happens. Lub happens when your bicuspid valve closes. Dub is where your semilunar valve closes. You only hear noises when stuff closes. You can know when these two are bad if you hear a lub shh, or a dub shh, or a lub dub shh, or something along those lines. If you hear a shh, like a whoosh, it means one of the valves is no good. So we don't want that. Moving on, we're going to talk about this over here. This is a cardiac cycle. It looks kind of weird, I know. We're going to start from the beginning. So here is kind of where you start. This is going to kind of be going over pressure and volume in your kind of like left ventricle and aorta kind of area. You start here, this is your left ventricle, you can start about 50 milliliters, that's for your bottom. Not really normally you wouldn't go any lower than that. You're going to kind of, your valve's going to open up leading from your left atrium to your left ventricle. Blood's going to pull in, and then that valve is going to close. Lub. Here is what we like to call preload. Preload is pretty much the volume of blood in the ventricle that is needed to contract. You need to have a certain amount before it will contract. If you don't, you're not going to really be able to push out blood through your aorta and your semilunar valve. So we can't have that, because that wouldn't be good. So now we are going to start constricting that ventricle until we build up pressure, and then our semilunar valve opens. And here's what we like to call afterload. Afterload is, let me get a better description of it than I can say. Afterload is the pressure to make, how much pressure is needed to have the semilunar, it's pretty much how much pressure is needed for the semilunar valve to open and for blood to leave. It's kind of self-explanatory. Then you get to about halfway here. All the blood is out of your ventricle. You don't really have anything in there. Now you're kind of closed, and you're going to close that valve. There's nothing keeping it open, and that's your dub. And we go back down. We lost all that blood. It's gone. And this just happens in a cycle, over and over and over and over and over and over. You can tell how much blood you're pumping by your stroke volume if you have a very large contraction or a very small contraction. It's not, you don't have to think about it this way, you can think about it down here. If I have 40 and 130 as opposed to 50 and 120, I have a higher stroke volume. Okay, it's a little bit interesting, but it's not too bad. This line here is representing inotropy. Inotropy is pretty much the strength of your contractions. And at this point where it meets where it goes here, you are just pretty much just saying at this point there's no pressure on your ventricle walls. It's not really too crazy. You wouldn't really need to know that, because I don't think you'd ever get to that point unless you're dead. And let me say one thing. In between these 2 points, between the part where you have your lub or your bicuspid valve closes and when your semilunar valve opens, this is called isovolumic contraction. So the same volume. You have the same amount here and here, but you're still contracting, you're building up pressure. It's nothing too complicated, really. There's just some complicated words. I believe that's pretty much everything. Thank you, and I'll see you in the next video. Hi, my name is Jack Corlius. I'm doing the third part of the third essay for Bio 461, and today we're going to be talking about inotropic, chronotropic, and lusiotropic pathways for acetylcholine and for neopinephrine or the catecholamines. First, there's 3 different pathways that we're going to be talking about. There's positive kind of chronotropic actions of catecholamines, such as neopinephrine and other things, and then there's negative chronotropic actions for acetylcholine. So we're going to start off with the positive, plus sign. I'm not going to draw it all out, but I'm going to put my First things first, we are going to stimulate our beta-1 androgenic receptor, and this is going to in turn increase adenylyl cyclase. This will increase cyclical AMP, P via the HCM receptor. And this can do 2 things. This can cause an influx of N of sodium, and this will in turn have we're going to see a steepness, we're going to have a higher slope of decay of membrane potential. So it's going to increase a little bit. this will increase your heart rate, because this will make it easier to depolarize and reach your threshold so you can go through an action potential. The other pathway, we can increase our pKa, and this will in turn increase our cyclocolane, cyclical calcium, influx, and our reticulum. We go two ways from here. Okay, we are going to have an increase in steepness of our slope of our depolarization. If we do this, it will make it easier to depolarize, so we'll have increased heart rate. The other way we can achieve this is by having the threshold more negative. And that will make it, you don't have to go as high to trigger an active potential, so you'll also increase heart rate. Let me just put an HR. HR stands for heart rate in this. So that is how we can positively impact it. But how can we negatively impact it with acetylcholine? Put a negative. This one we are going to go through our muscarinic 2 receptor. And there are 2 ways that we can do this. Well, not 2, sorry, 3. The first one is by triggering our beta gamma subunit. This is a heterodimer, so there's 2 subunits here. It's going to trigger them both. And this will open up our GERK channel. GIRK channel. And when we're doing this, the G protein is becoming activated, and it goes and activates the internal K channels. So we're going to have an influx of calcium, which is going to in turn, this will increase our membrane potential. Which will in turn increase heart rate. Similar to what we're seeing here. Actually, put that wrong. It's going down. Nothing too complicated, we're just going through another way. The other way, these next 2 ways revolve around the alpha subunit. And they both are going to be stimulating a decrease in our cyclical AMP. They're both doing the same thing. This is where they become different. This pathway, you're going to see a decrease in our channel for our F channel. And this will give us like a flattening slope. We're gonna just kind of see on a little graph. Instead of something like this, we're gonna see something more like this, which is gonna make it easier. Not easier, it'll make it harder to achieve our heart rate. Oh, so sorry, I put these the wrong way. Increase membrane potential, decrease heart rate. And while we're doing this, this is gonna make it much harder for us to achieve an action potential. So we're not gonna have as many, so we're gonna have a lower heart rate. On this one, similar, we're going to be stimulated, we're going to downregulate our calcium channels and make our threshold more positive, and this making it harder for us to achieve an action potential is going to increase our heart rate. All these are just going around different ways of increasing our heart rate by making things easier, bringing them closer to threshold. These ones are making it harder by going further away from threshold or something along those lines. that's those big ones. Now we are going to move into our androgenic receptors. These get a little bit more interesting. Androgenic is your beta one. I'm not going to draw all this out because it would take an hour, so I'm just going to give you the brass tacks. We are going to activate our alpha 1 subunit. From here, we can go to one of two places. As we saw, we're going to increase our adenyl cyclase, or we can interact with our alpha 1 receptors via our L-type calcium channels. Yeah, I'm going to write that here. Alpha 1 L-type calcium. Okay. When we're going this pathway, we're going to open our calcium. We're going to have an increase in our opening of our calcium channels. So what does this do? We're going to have more calcium. I know, shocking. And if we have more calcium in our cell, what did we learn about with our regular sarcomere? Our sarcoplasmic reticulum is going to have a higher release. If we have more of a sarcoplasmic reticulum release, we're going to have more contractions. Or not more contractions, we're going to have a higher strength of contraction. Now, that's one way we can increase our contraction, but there's two other ways that we're going to increase our contraction, and there's two other ways that we are going to shorten our duration of our contraction. All four of these, and we're going to go down a little bit more. We're going to have, since we're increasing cyclase, we are going to increase our cyclical AMP, which is going to in turn increase our protein kinase A. And from here is where we get a little bit more interesting. All four of these steps revolve around phosphorylation, but they each phosphorylate something else. So I'm just going to write phosphorylation. Okay. These two will be how we can increase our contractions, like increase how strong they are. These two will be how we can decrease the duration of those contractions. They're not necessarily working in opposition. They're at different points. The first one, we are going to be phosphorylating that calcium L channel. So it is going to be open. If we do that, what's going to happen? We can kind of see what's going to happen. We're going to have increased calcium, which is going to increase our sarcoplasmic reticulum to release, and then we are going to have an increase in contraction. So nothing too crazy. It's pretty much a different way of doing that. Nothing crazy. This one we're going to be phosphorylating. I'm going to draw little lines between here so we don't get confused. We're going to be phosphorylating our rhinodyne receptor. Rhinodyne receptors are, I believe, the second receptor. You have two different channels on your sarcoplasmic reticulum. This is one of them. If you do that, we are going to be able to dissociate calcium. If we do that, there's a little calcium that sits in there, and this kicks it out like your outgoing roommate it wants to drag you out to go and do fun stuff. If you do that, we are going to have an increased, we're going to be able to increase the time that our rhino event is open, increase open time. We do that, higher sarcoplasmic reticulum, increase to an increased duration of our contraction. Now here's where we get a little bit more interesting. I'm going to do, for this one, we are going to phospholize tropocine, troponin. If we do that, we're going to increase the rate of kicking, increase the rate of reabsorption of calcium, reuptake. If we do that, we're not necessarily going to prevent contractions, but we are going to increase the speed of relaxation because we're not going to have calcium there and our myosin heads can detach. And if we are increasing our speed of relaxation, we're going to increase our duration of contraction. This next one is we are going to be phosphorylate, I believe. Something phospho. To do that, we are going to increase our circuit pump. Our circuit 2 pump, to be more precise. If we do that, similar thing here, increase reuptake, increase relaxation time, and decrease duration to concentration. Decrease duration of contraction. These are 3 ways that you can affect the muscles in your heart. Heart muscles are very similar to any other muscle. You have cells, T-tubules, you got your sarcoplasmic reticulum that stores calcium. You are going to open up channels here that go in effect here. They release calcium, and you're going to have your sarcomere, which does what it does, goes through the cycles, and it can pull and stretch. And that's how we're doing it. The only thing that's different, it's smooth muscle. Well, it's its own kind of muscle. It's cardiac muscle, and it's happening faster. We reuptake it because we have pumps here, our circuit pumps, and they help bring calcium back in to the cell. And we have ways of kicking out sodium as well that we talked about in our other videos. I believe that's how we can get the movement of calcium in the system. Thank you so much. Hi, my name is Jack Corlux. I'm going to be doing the second part of I think this is the 4th part of the 3rd essay for Bio 461. I don't have my questions up, but I know they are. So first things first, we are going to draw out a capillary bed. It's going to be big, so bear with me. It's wonky, I know, but it works. So in our capillary, we have a precapillary sphincter. This is to help control blood flow and other things there are. I believe we have pancreas that help control this. So I don't think we need to go too into depth on that. In our cell, or in our capillaries, we have just regular hydrostatic pressure. We're just gonna abbreviate that to HP. We have a lot of stuff can go in and out of our capillaries, like glucose, sodium, chloride, potassium, and of course water. But one thing that can't is albumin. So albumin can't really go in, but our hydrostatic pressure wants to go out. We have things in here that can go out, so they're gonna go out. And the pressure of that is about 30 millimeters of mercury. Since we have stuff in here that can't come out, what's gonna come in? Water. 2O, and about that, the pressure of that will be around 28 milligrams. And we call this BCOP. All these things have names, but that's just so much easier if you abbreviate them, so that's what I'm gonna do. There's gonna be some stuff that comes out like that glucose, that sodium, the potassium, all that good stuff. And that is called your ICOP, and that is around 8 millimeters of mercury. We have stuff out here like proteins and stuff like that, like cells, like mast cells, stuff like that, that want these, and they're gonna get those, and that's how we can get them. We have one more thing out here, and that is a mast cell. Mast cells are kind of, not mast cell, lymphatic vessel. Lymphatic vessel. And these help move lymph and things around. These have little sphincters in them, so they prevent backflow, and they have a negative pressure of millimeters of mercury. So they're sucking things up. It's like a vacuum. And we can put all these together to find our net pressure. We don't need to quite do that yet. Or I don't think we need to do that for this. Now, we want to figure out like what are some ways that this can respond to with edema. Edema is when you have too much fluid. Like how does that affect here, or like how does that happen? So a pretty easy way, like a pretty common way you do this is when you work out. When you're working out, and you're pushing yourself, you are going to be doing micro tears in your muscle fibers, and that's going to cause the liquid to spill out, and you're going to be doing little tears. Tear, 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 tear. There's tears. Fluid and a bunch of other stuff. Your albumin is gonna want to go on out. So we have cells in our body, mast cells. Mast cells are immune cells. These immune cells have something called histamines in them. They're supposed to be anti-inflammatory. They're inflammatory. And all this stuff's coming out is going to trigger these histamines to release, and that can cause you to become inflamed. That's like why if you work out a lot, or if you run, or if you roll your ankle or something like that, that's why you get all puffy. You get puffy because of this stuff going out and because of your histamines. Your histamines are going to make it inflamed, very tender. We can talk about some of these things. So we have water that's going to want to come out now because all this stuff that was in here, it's outside now. So there's not a lot here. Our hydrostatic pressure, though, has dropped to around 10. Everything else is about the same. Our B cup is 28, but our I cup is 8. And our -3. Our lymphatic vessels. So everything's about the same, but this is what happens when you have edema or like something breaks in your vessels. You can get this just like from working out or something like that, or because you put a lot of blood in there and sometimes it will cause bursts. A common way that we can find, or like we can control like our sphincters and our vasodilation and our heart rate is through something called baroreceptors. And this is your aortic arch. You have these three big arteries that come off of it, and you have your top right here. In your top of your arch, you have these cells, these squiggles, here, and here are all baroreceptors. They are stretch receptors. Let's go to the spinal cord, right above your pons. I believe right above, yeah, your spinal cord, your pons. And this sends a signal to stimulate or control your central nervous system or your sympathetic or your parasympathetic nervous system. And that can control your heart rate. That's why you can do cautery massages if you have somebody that has really high blood pressure. You can massage these arteries here because there's vasorestrictors here. They are going to stimulate because you're stretching on them, and it's going to tell your spinal cord, hey, high blood pressure. We need to turn it down. And it's going to lower your blood pressure. It's not a cure, but it's a way to kind of help a little bit. Now, there are two things that I want to talk about really quick. Myogenic receptors. We have autoregulation and myogenic, autoregulation, and pancreas. Myogenic autoregulation is kind of something like the baroreceptors. You have smooth muscles, and these things, they're not quite like them, and they contract when you stretch them. And they send a signal to a place where they can release something or do something to reduce blood flow. Pancreas are similar, but they are chemical regulation using signaling to help do that, like I believe nitric oxide and adenosine, adenosine are popular ones used for pancreas. There's two different ways that there's two things that we want to talk about: active hyperemia and reactive hyperemia. Active hyperemia is a little different. You're going to be replacing something in your blood, you're going to be releasing a factor into your blood, which is going to decrease metabolic blood flow. Your metabolic blood flow. So it's going to vasoconstrict some. You're going to be lowering your blood pressure. Both of these are going to be in response to high blood pressure or like too much blood in an area. The other one is reactive. When you're working things out, you're going to be using oxygen. You're going to produce a oxygen-poor, carbon dioxide-rich environment. You're going to be using that nitric oxide and the adenosine, I believe it was, adenosine. And that's going to trigger things in your bloodstream to help raise your blood flow, which can help push out the carbon dioxide and the oxygen and provide nutrients to your cells. I believe those are the big things that we wanted to talk about with this essay. Thank you so much. You have a wonderful night. Hi, Jack Corlogue is going to be doing the fifth and final part of the third essay for Bio 461, and today we're going to be talking about hemoglobin and blood. So first things first, if you take a sample of your blood and you centrifuge it, or you just let it sit, you're going to have your blood in three layers. First one's going to be your red blood cells, or the C, and that's going to be about 45%. Here is your white blood cells, and this up here is your plasma. And your plasma is made of three things. It's made of albumin, globulin, globulin. And fibrinogen. Whereas with your red blood cells, mainly what you have in here is just hemoglobin. That's the primary thing that helps carry your oxygen. Hemoglobin is a pretty interesting mechanism. It has an iron atom in here. It's got two things branching off. Two amino acids that hold it in place. This iron can hold 4 oxygens. It has something called cooperative binding, so once the first oxygen binds, it can bind the other ones much easier. So it adds with each oxygen that's bound to it. For white blood cells, it has a lot of things in your plasma. You can kind of see the charge of what these are if you did an electric gel phoresis. You have a big hump, 2 ones, and a kind of big one here. This hump would be your albumin. This one would be like your alpha, beta, and gamma subunits of parts of these molecules. So one of the big things that we want to go over is you only have red blood cells in you for like not very long. I think a little over 100 days, if I'm not mistaken. So how do we recycle that? Because I believe birds, when they are going through it, they just poop it out. When they don't like it, they get rid of it. But we're a little bit more complicated because we were created in God's image. So we're going to go through the process of how we break things down. So we have our hemoglobin, and through a series of reactions using enzymes that are just found in blood, we are going to go down and we're going to turn this into biliverdin. Biliverdin has got a blue or bluish green color to it, whereas needs red. And we're going to undergo another reaction that will turn this into bilirubin. And bilirubin is kind of yellowish brown. And then we're going to transport this in the blood. And this is going to be bound, bilirubin is going to be bound to albumin. It's found in our plasma, and that's how we're going to be transporting it. Normally it's a non-polar molecule, but we need something to kind of transport so it doesn't get sucked off. So that's what we have albumin for. And eventually we go to the liver. And the liver filters things out, but the main thing that it does is it puts this sugar called, I believe it's glucuronic acid. On our bilirubin. And then from there we go to the gut. Once we're in the gut, the bacteria, the natural fauna and flora in our gut is going to munch up that sugar, the glucuronic acid. So we're kind of breaking it down. We're converting it into something else. And then we get to, once that's eaten that acid, we are going to convert our bilirubin into something else, and that is going to be our I'm not very good at pronouncing this, bilinogen. And from here we have a conundrum. Not really a conundrum, it can go two different ways. 50%, it's going to be split 50-50. So 50% is going to go back to our blood. Back to our blood, to our kidneys. And it's converted into urobilin. Urobilin's got a yellow color, and that is excreted through urine. The other 50% is a little bit more easy. It is converted into stereobilin. And then we get rid of that through our feces, and stereobilin's brown. That's kind of the big things here. It's a little bit more complicated, but it makes sense. Your body's a finely tuned mechanism. You're trying to conserve as much as you can. It costs energy to make things, so we want to have a as small effect as we can on our body, and how we can kind of recycle, reuse, and get rid of our things. Like the iron in our heme, it's just going to keep getting used up. We're never going to get rid of our heme, never going to get rid of that iron, because I think it's like when people say, oh, you had to eat a lot of iron. None of that really is getting into us. There's a little bit, yeah, but it's like under 10% of your dietary iron goes into you, like is used in heme. So that we really don't need to worry about. We're trying to keep it in us. Similar to calcium, but we use more calcium. So now we're going to talk about homeostasis. There's three ways that we can achieve homeostasis in our body. And these all kind of have to do with clotting. The first one's vasoconstriction. It's pretty simple. Got our little vein right here. Something happens to our vein, we have stuff come out. Our smooth muscle undergoes I believe it's called a vascular spasm. And it stretches really tight, and we don't lose as much blood. We have a limited blood flow to that area, so we're not losing as much. Well, the problem with vasoconstriction, it's not permanent. You can't just think about it like flexing. You can't like keep this immaculate pose like myself forever. Eventually you have to release. Even with smooth muscle, even your sphincters, eventually those sphincters are going to have to open. They can't be closed indefinitely, and you need blood supply. So the other way we do this is called platelet, I can't spell, aggregation. And you're like, oh, platelets, I've heard of those, they're in the blood. They are. So we're going to draw a bigger vein here, so I don't have to reach up. It's a big vein, and we have an injury. In our cell, like in our vascular system, we have two kinds of cells. I'm just going to draw these cells here. These are your epithelial cells, and you have something here called your basement cells. Now, in your blood, we have platelets that are going to be constantly in them. Here, like those are pretty big, but you get my point. They're in our cells, and they're not active. We're not going to activate. If you activated them in the middle of your vein, you'd get a pulmonary embolism and die, or a stroke, or a heart attack. It wouldn't be good. And these little guys have I believe the term is a GP1D receptor on them. And these receptors bind to this little protein here Now, we also have something else in here. I'm gonna grab a different color real quick. These guys, and these are fibrogen. These receptors can bind to these or these, but they can't bind now because there is nothing. They're not activated. You have to, your platelets have to be active in order for you to be able to bind. But if we have damage here, VWF loves to bind to our basement cells. So we have stuff like that here. And our platelet's gonna go and it's gonna say, hey, I kind of want to be close to this. And that GP1D receptor gets on there, and as our blood flow is pushing this way, it stretches the platelet. And I guess you could say stretching or pulling, and that activates the platelet. And this activated platelet looks something like this. I'm going to use some artistic liberties here. Okay, that is our activated platelet. I'm going to go through some of the anatomy of the activated platelet real quick. Here's our activated platelet. We have a little guy in the center here that is called your TXA2. And this is a thromboxane. I don't know, it's there. And we have two kinds of subunits here. They look the same because I'm using black, but they're different. We have dense granules. That are receptors for ADP, and calcium. And then we have alpha granules. Receptors for PDGF and for two proteins that are just called factor 10, factor 5, and factor 7. And then we have those receptors that we talked about. These ones are also called, once they're activated, they're called GP2B3As, and they're fibrogen receptors. That becomes important because once these bad boys are activated, this guy's bound to this guy's bound here, but fibrogen's going to come and it's going to bind here, and other places as well. And these other platelets are going to be going, I can bind to fibrogen. I'm going to bind to that. I'm going to get close and bind to that fibrogen. And once they do, they activate, and the whole cycle continues. And this is how we get the platelet clotting. You get a little platelet patch here, and eventually your thing can heal up, and you're not bleeding anymore. And that's good. We like not bleeding. The third and last one is called coagulation. And big here too. So there's three kinds. I'm gonna really go into depth on two of them. The third one is not too different. The first one is intrinsic and extrinsic. So once we have the same kind of thing happening, we have our epithelial cells, we have our basement cells. Everything. And once we are open to the things out here, we start a cascade of proteins. There's not really much different here except for by what's activating it. Intrinsic something from the inside is activating it, and extrinsic something from the outside is going to be activating it, and you use different proteins. This one we have factor 13, and it goes into an activated factor 13, and then we have factor 11 to an activated factor 11. No, it's 9. That one is converted into a 10. 10A. A just means activated. This one's converted from a 2 to a 2A, and then from a 2 to a 1 to a 1A. And once we get this protein, we fix these. Now, the big difference between this and intrinsic and extrinsic is just what starts the cascade. For this one, we start with factor 7. Factor 7 is factor 7A. And we have 10 here. Yeah, and then pretty much from there, this little part right here goes here. We just copy and paste this over. The really only difference is we have a couple extra steps here. Now there's a third one called common. Common is a little bit more interesting. We are going to attach a factor 10 plus a factor 5 to a calcium and platelet. And when you activate those two, you are going to form a prothrombin activated complex that targets your this enzyme will find protein 2, and that converts your fibrogen to your fibrin. So it's basically a really easier way of doing this. You don't have to go through all the steps, but once you kind of start this cascade, there's really nothing stopping it. You're going to have all these things in abundance in the bloodstream, so common's a little bit harder to do because you have certain things that you need to have in the blood. Like if you don't have a platelet-rich environment or a calcium-rich environment, and you don't have these two, you can't undergo common. Let me put that here. But with intrinsic and extrinsic, all these things are going to be in high prevalence in the blood. So you are pretty much, it's going to happen. It's not an if and. There's no if and or buts about it. This one, a little bit more so. I believe that's it for this final one. I'd like to thank you all for joining me. Thank you.